Hello everyone, I am Furkan Ozden from Bilkent University, and today I'm going to present our newest research on polishing copy number variation calls on exome sequencing data via deep learning. Let's get started. Gene copy number polymorphism in a population due to deletions and duplications of genomic segments substantially drive genetic diversity, affecting roughly 7% of the human genome. This class of structural variations, named copy number variations, shortly CNVs, are mainly categorized as deletions and duplications. CNVs have been associated with several genetic diseases and disorders, such as breast, ovary, and pancreas cancers. Karyotyping and microarray analysis have been the standard clinical testing for disease-causing CNVs for many years. But lately, high-throughput sequencing has all but replaced these techniques with the ability to theoretically capture all forms of genomic variation. Many CNV detection algorithms have been successful on whole genome sequencing data using different sequence signatures such as read depth, discordant paired and read mappings, and split reads. Overall, whole genome sequencing is a convenient resource for CNV coders as it provides uniform read depth distribution. On the other hand, accurate CNV detection algorithms on whole exome sequencing data has mostly been lacking. The algorithms which call CNVs on the whole exome sequencing data have notoriously high false discovery rates, reaching up to 60%, which renders them impractical for clinical use. This is mainly due to several problems associated with the whole exome sequencing technology. One major source of such problems is non-uniform read depth distribution among exons. As we see here in this IGV screenshot, the read depth distribution for whole genome sequencing reads are uniformly distributed among exons, unlike the situation for the exome sequencing data here in the bottom part of the screenshot. The non-uniformity of the read depth signal is caused by biases in sample batches, GC content, and targeting probes. This is unfortunate as whole exome sequencing data is 10 times smaller and it costs 3 times less compared to whole genome sequencing technology, which makes it highly abundant and a common choice for analyzing complex genetic disorders. Therefore, currently such a rich resource of large-scale whole exome sequencing data cannot be fully utilized to investigate the contribution of copy number variation to disease etiology. Here we present the first of its kind exome CNV call polisher named dcont to improve the performance of any off-the-shelf exome sequencing-based germline CNV detection algorithm. dcont is a deep learner that utilizes matched exome sequencing and genome samples present in 1000 Genomes project. We designed dcont to specifically learn the association between calls made by a whole exome-based CNV caller and its corresponding ground truth calls generated from genome sequencing data for the same samples. We designed dcont based on a bidirectional long short-term memory deep neural network architecture to allow our model to learn and correct the CNV calls from a third-party exome CNV caller. Both exome and genome samples are sequenced using Illumina Genome Analyzer 2 and HitSeq 2000 platforms, and reads were aligned to GRCH38 using Burrows-Wheeler aligner. As we see, dcont is a multi-input neural network designed for polishing and improving the performance of the exome-based germline CNV coding. dcont's pipeline for the categorical CNV prediction case can be divided into 3 main building blocks, and it can easily be modified for integer CNV prediction case just by changing the softmax activation with some linear activation along with a suitable loss function. Firstly, dcont has a data preprocessing step that extracts the read depth information for genomic region of interest. It also normalizes the read depth sequence and acts as a regularizer for the model. Resulting read depth sequence is padded to the length of the longest sequence, and then it is masked. The second part consists of a bidirectional LSTM network that inputs the read depth sequence and extracts the required encoded features. The second part of dcont's architecture has 128 neurons in each LSTM direction, and it is followed by a batch normalization layer. The third part consists of a two-layered fully connected neural network that inputs the encodings predicted by bidirectional LSTM, and these are concatenated with the prior CNV prediction of the CNV caller. This subnetwork has 103 neurons in total and uses ReLU activations for each neuron except the softmax activations. We use cross entropy as our loss function in the model, and DCON's architecture has around 160,000 learnable parameters in total. For the tools which predicts the existence of a CMV event are evaluated with respect to duplication, deletion, and overall CMV call precisions. DCONT improves the performance for all tools in all settings and results in drastic improvements. Different shades of gray represent different tools, and the attached black bars represent the DCONT versions of those tools. The largest gain is in duplication call precision for Conifer, which is improved by 2 folds, that is from 24% to 75%. And the largest gain in deletion call precision is again obtained for Conifer, which is improved by around 50%, which corresponds to an increase from 45% to 68%. Also, the overall precision is improved by 1.6 folds, which is the increase from 27% to 71% for Conifer. This improvement is especially striking as Conifer is a relatively conservative tool, and compared to other algorithms, this tool seldom makes calls, despite relaxation of its parameters. For XHMM, we observe 1.4, 1.7, and 1.5 fold increases, which correspond to a 20% increase in duplication call precision, a 29% increase in deletion call precision, and a 24% increase in overall call precisions. We see a similar trend here for codecs. As for the tools which output integer copy number values, namely Control Freak and CMVKit, we evaluate their performance against absolute error. For Control Freak, we consider around 20,000 CMV events, and DCONT improves the absolute error in 74% of the test samples, for an average absolute error improvement of around 47%. And DCONT deteriorates the performance in around 25% of the test samples, for an average absolute error deterioration of only 1.2. Unpolished Control Freak predictions have a Spearman correlation coefficient of 0.22 with matched ground truth copy numbers. DCONT polished predictions have a Spearman correlation coefficient of 0.57. As we can see here in the distributions for the box plots, similar to Control Freak, DCONT polished CMV predictions highly resemble the distribution of the ground truth calls for the CMVKit case. In order to test the polishing performance of DCONT, we also use a highly validated CMV call set published by Chason et al. In 2019. This dataset contains the whole genome-based CMV calls of 9 individuals selected from the 1000 Genomes Project samples. And for these samples, a consensus call set is obtained by authors using 15 different whole genome-based CMV callers with comparisons against high-quality structural variants generated using long-read sequencing with single base pair resolution. DCONT improves the performance of all 3 algorithms in all comparisons. And the most substantial improvements are observed for Conifer with up to 31% improvements. For XHMM and codecs, we see consistent improvements in performance reaching up to doubling the precision performance. As we have already said, we obtained the training dataset from the 1000 Genomes Project database, and the exome sequencing component is produced using Illumina Genome Analyzer 2 and HitSeq 2000. The data from these platforms is abundant and sufficient training data sizes can be met. However, for users using other sequencing platforms, it might not be possible to train DCONT due to lack of matched exome and whole genome samples. Therefore, we evaluated whether models trained on the available 1000 genomes data can be used to polish CMV calls made on exome samples obtained using other sequencing platforms that have not seen by DCONT during the training phase. We obtained the exome data. We obtained the exome data for this test using 4 different sequencing platforms: NovaSeq 6000, HitSeq 4000, BGISeq 500, and MGISeq 2000. Even though DCONT has not seen the read depth information or the CMV event on these sequencing platforms, it still can generalize from the training on the 1000 genomes dataset and still can substantially improve the performance of XHMM, Conifer, and Codex. We observed improvements in 34 out of 36 tests, and the most substantial improvement is observed for Codex that corresponds to an average 2.6-fold increase in performance, and this even exceeds the testing performance on the same platform as training. For XHMM, the performance is improved 10 out of 12 tests, reaching up to doubling the performance in overall precision performance for BGISEq and MGISEq platforms. For NovaSeq 6000 and HiSeq 4000, the performance deteriorates in duplication precision. However, XHMM makes a few duplication calls on these platforms. While Decont keeps the true positives, it adds a few false positives, and this results in the performance decrease in these settings. We note that the improvements provided by Decont on the BGI and MGI platforms are important, as these systems belong to a completely different manufacturer. And since these platforms are expected to have different systematic biases in read depth distributions, compared to the training data of Decont, of course, we also expect a lower testing performance. Yet, Decont is able to generalize well and consistently proves to be useful across a diverse set of technologies. Overall, the performance is on par with the tests obtained on the platform settings, identical with the training phase. Polishing procedure consistently improves the performance in a platform-independent manner. A distinct Decont model is trained for every exome-based germline CMV caller. This makes sense as the call regions and numbers are substantially differing among algorithms in their recommended settings, and for this purpose we check if a Decont model trained using calls made by one algorithm can be used to polish the calls made by others in the absence of a trained model. We use the same Decont models trained for XHMM, Conifer, and Codex on 1000 genomes project data, and for each tool-specific Decont model, we polish the calls made by others on samples not seen during training. For instance, we polish the calls made by Codex using the Decont model trained on XHMM calls, and this experiment results in 6 tests, and each test has 3 metrics, resulting in a total of 18 performance results. We observe that Decont improves the performance metric in 10 out of 18 comparisons, and XHMM trained Decont consistently improves the other tools' performance in all metrics except deletion precision when polishing calls were reported by Conifer. While arguably it can be used to polish calls generated by other tools, a Decont model trained on the calls of to-be-polished exome-based caller is suggested. As we see, the improvements in the discussed performance metrics are larger in this case, which is expected. In this work, we show that Decont can improve the duplication and deletion CMV call precision of the state-of-the-art exome-based CMV calling algorithms by approximately 3 folds and 2 folds respectively. We noted that the performance gain is consistent among CMV callers that output integer copy number predictions and categorical predictions. As the training phase of our algorithm is offline, polishing procedure is memory and time efficient and takes only a few seconds on average per sample. Furthermore, we show that the trained models are universal in the sense that they are sequencing platform, capture kit, and CMV calling algorithm independent in polishing performance. So thank you for listening to me, and a few quick notes. Decont is implemented and released at GitHub. The link for the repository is given here, and all custom Python scripts that were used to generate matched genome and exome CMV data are also available on the GitHub page. The scripts we used to generate the data for all figures and the figure and table generation codes that were used in the manuscript are provided at Zenodo, and the link is given here. So thank you so much for listening. If there are any questions, I am more than happy to try to answer your questions. Thank you.